Excellent. All right, I think we're good to go. Are you guys seeing a full screen right now of PowerPoint stuff?
Yeah, we are.
Perfect. All right, so we're good to go. All right, so good morning, guys. My name is Neil Balchan. It's really an honor to be here the second year in a row. I really enjoyed giving this talk last year, and it's just a great group and a great project that you guys have going. So thank you so much for having me back. So I'm a bit of a snake guy, I guess to put it lightly. And I've kind of titled today's talk, The Art of Being a Snake. And I think it's very vague, but it's also kind of intentional because snakes are, a very different creature. And I think there is a bit of an art associated with actually being a snake. So let me dig right in. So there's around 4,000 species of snake in the world. And that number goes up every day, right? We're continuing to describe species. But the thing that I kind of want to just drive home with this slide is that snakes are not boring, right? People often think of, a snake being this kind of creepy, slinky thing hiding in the grass. They're anything but that, right? They're this incredibly diverse radiation of living things that we have on the planet, right? So we have these kind of rough brown sidewinding rattlesnakes that live in some of the harshest deserts in the world. And these might represent, one extreme of snake diversity. We have these beautiful tricolored coral snakes, which are distributed throughout the New World tropics. So again, really beautiful snake, but totally different kind of end of the spectrum. Next up, we have things like garter snakes, which while they're very familiar and very common, can be beautiful in terms of their color and their patterns. And now if we think about snakes that are maybe a little bit more familiar, big snakes, right? So we have things like the green anaconda that can get very, very heavy. We have snakes like the Burmese python that can get very, very large and very heavy. And these are snakes that are, huge and charismatic that we're very familiar with. But then there's a lot of snakes that we're very unfamiliar with, right? And the guy on the bottom right is part of a group of snakes called the shield tails. And they're named that because their tail is hard and it's kind of fused into this shield. And again, that's a snake that really isn't familiar to a lot of people, including people who work on snakes. So the thing that I just want to drive home is that a snake is not a snake is not a snake, right? There's a lot of different snakes out there, and these guys are all being a snake, for lack of better terms, in a different way. So super cool group of animals to kind of pick apart and work on. So a little bit about myself. So I grew up in central Canada in a city called Winnipeg. And I think I, came out of the womb, I guess, for lack of better terms, as a snake guy. And here's a picture of me with my family as a very, very young kid. So the top one is, me being held by my grandpa, and then my grandma and dad are kind of adoring over me with the snake there. And there's me below with my mom. And the thing is, even as a young child, these snakes were just fascinating to me. And I was enamored by this snake diversity. And unfortunately, that bug didn't really go away, right? And the nice thing about going to school and continuing on through university is you're allowed to chase whatever weird thing keeps you up at night, right? And for me, that's snakes. So here's just a couple pictures from Southeast Asia. So on the left, there I am in Laos with a really, really cool viper. And on the right, there I am in Thailand with another really cool viper. So the nice thing is you can continue to do this in science. And that's one of the nice things about science. So I'm going to split today's talk roughly into two subject areas. And these give kind of a good overview of the research that I've done so far and the research that I continue to do and kind of want to move into. So the first segment, we're going to call this biochemical ecology of venomous snakes. I know that's a lot of big words, but just bear with me here. All right. So what is biochemical ecology? Well, in short, it's really just regular ecology, but involving chemicals. And when I say chemicals, we're talking about poisons, or toxins, or venoms, things like that, right? That interact between one or two or more organisms. So if you think about examples of this in the normal world, right? The monarch butterfly, this is a good example. Beautiful, brightly colored butterfly, and it's actually toxic. So its ecological interactions are kind of dependent on this toxin, right? Predator-prey interactions. The frog below it, that's a poison dart frog, something, again, that should be pretty familiar. And these frogs are poisonous, right? So they have bright colors, they're poisonous, and different species will either avoid them or eat them, with a knowledge of this toxin. And then finally, the newt garter snake example is really cool. So here we actually have a newt that is super toxic, and then a garter snake that has evolved really high resistance to that toxin. So again, right, toxin and toxin resistance kind of working together ecologically. So really, really cool ideas going on in this field. Now I want to pivot a little bit here, and I'm going to switch to a quote because most people don't like snakes. So we're not going to pretend that's not the case, right? So this guy here is A.F. Scutch. He's a North American ornithologist. He's a bird guy. And he went on record and said that the serpent crams itself with animal life that is often warm and vibrant to prolong an existence in which we detect no joy and no emotion. It reveals the depths to which evolution can sink. So this guy didn't like snakes, but I think his statement is factually wrong on a couple of accounts, right? Snakes eating things isn't really this cold, calculated deathly pursuit. There's a lot we can learn from it. And I think when we think about that biochemical ecology, there's even more we can learn from it. All right, so venoms. What are venoms? Well, venoms are this complex cocktail, if you will, and they're a mixture of proteins, peptides, and enzymes. And they do a bunch of different things for our snake friends. So one of those might be digestion, right? And there's some evidence that rattlesnakes especially may have some digestive function to their venom. The jury is still kind of out on that. Now, another potential function of venom is defense. And you can think about the spitting cobras, right? They're going to be one of the most charismatic members of this group for that. And these snakes can actually propel their venom through the air, right? They can spit venom. So defense. But the most important function of venom, without a doubt, right, is going to be prey acquisition. So you inject your venom into what you're trying to eat. That thing dies, you can eat it, right? Pretty important reason to have venom, and it's a pretty important driver of venom evolution. So let's start by understanding what snakes actually eat. Why might they have venoms that look the way they do? So what do snakes eat, right? Well, most people think that snakes eat things like this, right? So your normal snake might eat a mouse, maybe it's eating a rat. If things are getting really kind of exciting, maybe that snake will catch a bird, right? But again, this comes from our very familiar view of snakes. And we have to remember that the art of being a snake is actually quite complicated. So while these are normal snake foods, snakes will also do things like this. This is just a short video here. All right, so I'm not sure if that surprised anybody else, but it definitely surprised me. And what we're looking at here is a type of rattlesnake. It's called a Massasauga rattlesnake, and it's actually going after a centipede. And who in their right mind would have thought that a snake that, should get by eating mice and things like that, maybe lizards, who would have thought that it would be eating centipedes, right? So this kind of forces us to think a little bit more broadly. What are these things actually eating? And when we think about that, why might venoms look the way they look, right? What might be different in a venom that's kind of tailored for a centipede versus one that's tailored for a mouse? So weird little tangent, but I think the idea here is just to keep your mind open. Snakes aren't necessarily eating what we think they're eating. All right, don't need to watch that again. So let's think about a typical day in the life of a snake, right? You have to catch a mouse and eat a mouse. So here's how that goes. So you're a rattlesnake, you strike your mouse, you let it go. That's pretty typical. And then your mouse has a couple of options, right? It can try to run away. It's probably not going to work. It's got venom in it. It could try to hide. Probably also not going to work. Or it could do this crazy thing, which is actually resist that venom. So it could turn off that venom from its, very insides. And we do see this happen. And this is kind of a light bulb moment because it represents kind of, the mice winning in this venom versus lack of venom arms race. And again, the depths to which evolution can sink. I disagree. I think this is a really cool kind of phenomenon that we get to study. And this touches on this idea called the Red Queen hypothesis. You might learn about this a little bit in some higher level biology courses, but it's this idea that organisms that interact with one another are co-evolving. So when one critter does something, the other critter kind of does something back, right? And they do that to remain competitive in their natural environment and with respect to one another. So what does that mean, right? So again, let's think about things in the real world. So Daphnia, they're these tiny little water creatures, these tiny little crustaceans, and they co-evolve with their parasites. But if we get a little bit bigger, right, that garter snake and newt that we just talked about, this is one of these Red Queen scenarios, right? The newt is becoming more toxic over evolutionary time, and the garter snake is becoming more resistant to that toxin. So bingo, there it is. And I would say that venomous snakes and rodents do the same thing, right? So venoms of snakes are evolving, they're potentially becoming more toxic. And conversely, rodents have to be able to negate those venoms, right? So they're going to evolve in a direction that will hopefully make them more and more resistant to venoms. So here's an example of that, right? All right, and we see this, of course, in the wild. So the California ground squirrel, it's this beautiful little squirrel in the western US, and it exhibits a very high resistance to the venom of the rattlesnakes that eat it. So again, most of the time, right, it can probably get bit and shake it off, but this isn't an all or nothing event, right? Some of these squirrels are going to be less resistant or more resistant. Rattlesnakes may inject varying amounts of venom, so they do still get eaten, and that's still driving this continuum. So squirrel, good. But if we think about a different squirrel, this is a South African squirrel now. This particular squirrel does not have any venom resistance to the snakes that it co-occurs with. So if it's bitten, here by a Cape cobra or by a puff adder, it's not going to have any way to really neutralize that venom. So maybe we don't see it in all rodents. And that brings up an interesting question, right? Why do some rodents do this and then other rodents not do this? So again, this squirrel, thumbs down. And this kind of gets into a deeper area of thinking, right? So I won't bore you too much with this, but the whole idea is that everything is really complicated. And when we think about a venom, right, a venom is this cocktail. It's made up of a bunch of different things. And a rodent or whatever animal needs to be able to resist each of these different things independently, right? So just bear in mind, this isn't really a simple thing. It's actually a very complicated thing. All right. So I wanted to play with this a little bit, and I did that for my master's in Colorado. Colorado was perfect because we had two rattlesnakes. We had the prairie rattlesnake and the desert Massasauga rattlesnake, and these occurred kind of at a spatial gradient. So number one here on the map only had the prairie rattlesnake, and then number two on the map had both rattlesnakes. So that was kind of exciting. And then in addition to that, there were a bunch of different rodents that lived on this landscape. So all of these lived at both sites. So deer mice, house mice, grasshopper mice, and then a kangaroo rat, which was a really neat little rodent. So I was really curious about how things worked in this ecosystem, right? Who's eating who? While Massasaugas are a very small rattlesnake, they're probably only eating the two small mice, right? And then prairie rattlesnakes are much bigger. And because they're bigger, that means they can probably eat everything. So they're probably putting high predation pressure on all of the rodents in this ecosystem. All right, so let's think a little bit about venoms. So we knew a little bit about prairie rattlesnake venom coming into this already. And we know that the snakes actually have different venoms in different locations. So prairie rattlesnakes at site 1, they have what we call a high myotoxin, low SVMP venom. And just, quick and dirty here. So what that means is that this venom is paralytic. So you get bitten by it, you become paralyzed, but it's not as toxic, right? So it slows you down, it paralyzes you, but it doesn't necessarily break you down as much. So these snakes in the south are a different version. They're the second, option of that, right? So these are low myotoxin, high SVMP. So not very paralytic. They're not going to stop you in your tracks, but they are going to be very toxic to your body. They're going to break down your tissues. They're going to cause a lot of bleeding, very, damaging venom to whatever it bites. And what that means is that these look different when a mouse is bitten by them, right? So the snakes in the north, you get bitten by a northern snake, a site 1 snake. It's a much more, directly toxic venom, right? So it only takes 1.25 micrograms of that venom to kill a mouse. The one in the south, right, this number 2 venom, it takes twice as much venom to kill a mouse. So just things to kind of keep in mind for how these venoms might work on the landscape. All right, now just some quick hypotheses and predictions, right? So we think that venom resistance will probably correlate with predation frequency. So how much do you eat the rodent that we're interested in, right? And then we also think that venom resistance will probably be dependent on geography, right? So if you're a rodent at site 1 and you're a rattlesnake at site 1, you probably have co-evolved with each other in evolutionary time, and you should have greater resistance, right? Whereas the opposite, if it's a site 1, site 2, mismatch. All right, so how do we do this? So we started by collecting venom. So we went out to the field to both of these sites. We captured a bunch of snakes and we took venom from them, right? So the bottom image there is just venom being taken from a snake. Once that venom's out, it's really easy to work with. You just freeze it and then you dry it down and you can use it at any point you'd like to. It'll last pretty much forever. All right, and then we went ahead and we trapped some rodents. So we caught those with live cages and we moved them to the lab for the work that we were gonna do. All right, so just a little bit of methods, don't get too bogged down with this. But we did two main experiments or two main assays, right? And the first one is this one called an LD50 assay. What that is the median lethal dose. So all that means is that it's the dose of a venom that's actually required to kill half of a population. So if I have 10 mice, right, how much venom per mouse would be required for half of those mice to die? And this basically tells us how toxic a venom is. It's just a quick number for how toxic that venom is on that particular population of rodent or of animal in general. We used another test here. We call this one a metalloprotease assay. So metalloprotease is an enzyme in that snake venom, and that's what's really breaking down that tissue, right? Causing bleeding, causing tissue degradation, and we can test for that. So we tested for that in the lab, basically using like a test tube type assay. And all this told us is if the blood of a particular rodent had any protective quality against that enzyme, against that venom. So if I mix blood of, let's say a kangaroo rat with venom of a rattlesnake, is that blood actually going to protect that kangaroo rat any? So a little bit different way of looking at it. All right, and here's what we found. So don't worry too much about the graphs. These aren't super important, but a couple take homes, right? And the main one on this particular slide is that Weld County prairie rattlesnake, so that northern one, is more toxic to its co-occurring mice than it is to mice that do not occur, near it. So what that basically tells us is that in this particular case, right, so this prairie rattlesnake deer mouse example, it looks like these deer mice are actually losing the race locally, right? So the prairie rattlesnake at this particular site has evolved in a direction that makes it more toxic to its local mice than it does to these deer mice generally. So really neat kind of thing coming out there. All right. And again, those LD50 values really indicate that quite nicely. And if we think about some different rodents, so these are, again, deer mice, and this is that blood serum example, right? So thinking about how that blood is going to affect that venom. And we actually see a very weak inhibition. So the blood of that mouse isn't actually doing much to turn that venom off. We do see maybe a little bit more heightened inhibition here in this pairing. So this is a Lincoln County deer mouse to a Weld County prairie rattlesnake. So those mismatched mice. But again, right, nothing crazy going on here. This blood doesn't maybe have anything in it to do much as far as turning that venom off or detoxifying that venom. All right, so house mice, right? So house mice are not a native animal in the US. They haven't really been on our landscape for very long. They were brought here by people. So the question is, are these mice that are now living wild in the US, have they evolved at all? Are they, used to snake predation? Have they adapted to it? And it turns out that they haven't. So here we took wild house mice that we trapped in northern Colorado, and we took domestic mice, lab mice, and we tried them against prairie rattlesnake venom, that northern prairie rattlesnake venom. And what we see here is that these wild mice and these domestic mice do not have any difference in their susceptibility to the venom. So mice are not more resistant, lab mice, that is. All right. And then another little native rodent here. So this is a critter called the Plains pocket mouse. It looks, kind of like a little gerbil. It's a really cute little thing. And it's a small rodent. They're probably eaten by Massasaugas a fair bit. So we were wondering if maybe, hey, this is a really common Massasauga prey item. Could it be that it's resistant to Massasauga venom? And in fact, it's not. So we see, 18% inhibition and 15% inhibition from mice at these two different field sites. But we don't actually see any heightened inhibition where these mice co-occur with the Massasauga, right, which is Lincoln County. So this would suggest to us that we haven't really evolved in a direction of resistance against Massasauga venoms. All right, and then wood rats. So these are a really cool rodent. These are a big rat, and they build kind of a mound. It almost looks like a beaver lodge, but it's on land. So these are a critter that have been worked on, a fair bit in the past, and they have been found to be very resistant to rattlesnake venoms. So in our particular study system, we took these wood rats and we assayed them against Weld County venom, Lincoln County venom. So these are both prairie rattlesnakes, and we find very high inhibition, right? So with these Weld County venoms, 42% of that venom activity, that protease activity, is being turned off by wood rat blood. So that's a lot. And then in Lincoln County, 69% of that protease activity is being turned off. So if this rat, simplistically, if this rat is getting bitten by a Lincoln County rattlesnake, 70% of that venom is just not doing anything. So that's amazing, right? That's a really, really good adaptation for this rat to have. But then conversely, this rat doesn't inhibit any of the activity of this Massasauga venom. And that seems kind of odd, right? We wouldn't necessarily expect that. But when we think about a Massasauga, it's a tiny little rattlesnake. It's probably not eating rats. So this rat doesn't have any reason to evolve, resistance to that venom. All right. So a little bit more resistance data. Northern grasshopper mouse, really resistant all across the board. I'm not going to dig too deep into him. But the one really, really cool thing is these LD50 values, right? So this grasshopper mouse is bulletproof. And when we think about, a lab mouse, it can take an LD50 of maybe 1 to 3, let's just say. This grasshopper mouse can go up to 127.7, right? So it is, dramatically more resistant to snake venom than these lab mice are. So really kind of a little superhero mouse here. All right. Ord's kangaroo rats, same deal, right? 125 versus 1 to 3 range, let's say. So again, little rodent superhero. So while a lot of these rodents aren't doing much, some of them are doing some crazy things, right? And the grasshopper mouse and the kangaroo rat would be an example of that. All right. So with that said, conclusions. What does any of this mean, right? So lots of these rodents do have resistance to snake venom. Other rodents do not, right? And I think the take home here is that predator-prey interactions are complicated. There's just a lot going on, and it's hard to really account for all of that sometimes. Okay, so hopefully that didn't go too deep down the rabbit hole here. But the big picture of this kind of subset here is if we can figure out how rodents are doing this, right? How rodents are turning off these venom, cocktail ingredients, if you will, we can flip that back into human health, right? And snakes obviously are a big problem when we consider snake bite. So if we can figure out how to implement some of these strategies that our wild kind of mammalian counterparts are doing, maybe we can find a solution to better treat or to better manage snake bite, particularly in the developing world. All right, so what about the things that actually eat snakes? So we're going to pivot a little bit. We talked a lot about the things that snakes eat. Now let's talk about birds of prey, right? So birds of prey are potentially one of the biggest predators of snakes, maybe even globally. So raptor diets are poorly characterized, but we know that most species eat mammals and birds and things like that. But many do eat amphibians, reptiles, and invertebrates, right? And among that, of course, venomous snakes are important. So the images below just show 3 different birds eating 3 different venomous snakes. So the first 2 are red-tailed hawks. They're both eating rattlesnakes, so a prairie rattlesnake and a timber rattlesnake. And then the final picture shows a serpent eagle eating a cobra. So a lot of snake predation going on here. And we know that this is a thing, right? So we have opportunistic predation, as we saw the slide before, but then we have snake specialists. So things like the secretary bird, the seriamas, the snake eagles, the laughing falcons, these are birds that eat a lot of snakes. And these interactions are so kind of charismatic that we even see them in culture. Here's just a picture from the Mexican flag of an eagle, kind of sparring with or dueling with a rattlesnake. So we've been thinking about this for a while. So how do raptors avoid envenomation? Well, they could have physical mechanisms, right? They have things like feathers and really hard feet. These might be hard for a bite to get through. They have behavioral mechanisms. So a lot of birds of prey will do, some kind of display or some kind of dance to confuse a snake while they're trying to catch it. But there's also potentially these physiological mechanisms, right? So the things we just talked about with our rodents. So venoms, we did this already, just a quick refresher. And again, venoms are very complicated, right? So what do we know about predators that avoid envenomation or predators that kind of neutralize venoms? So we know a little bit. So cobras and honey badgers here, we see resistance. Rattlesnakes and opossums, we see resistance again. And then this terrifying little snake called a stiletto snake. With a mongoose here, we see resistance. And these are all various different toxins and toxin families. So pretty widespread things, right? But these are all mammals. And I want to really kind of indicate that these are all mammals. Most of the work we've done has been mammals. So what about cases where there isn't resistance, right? So again, we talked about that South African ground squirrel. Sure, no resistance, but again, a mammal. So I went ahead and I did some digging and I found some bird examples. And here's one right here. So a peacock, right? Peacocks love to eat snakes. And we found that they were not resistant. There's no resistance to these snake alpha neurotoxins in peacocks. But then it gets more interesting. So this bird, this serpent eagle, and this is a snake specialist, also does not have any resistance to snake alpha neurotoxins. So things are getting weird now. Maybe birds don't have resistance to snake venoms, right? So we wanted to look into that. And we did that here using our same system. So prairie rattlesnake, I was in Colorado at the time, and we worked on the birds that we had local, the birds that ate snakes, right? So these were the red-tailed hawk, the Swainson's hawk, the bald eagle, the golden eagle, and the great horned owl. So for this, we only worked with blood. We didn't want to have to euthanize any birds of prey, obviously. So we collected blood from a handful of birds at a raptor rehabilitation program. So 9 red-tailed hawks, a Swainson's hawk, golden eagle, bald eagles, and then great horned owls. And then we actually used chicken blood as a control. So same protocol, right? Test tube assay. We looked at how protective that blood was. And what we found, in short, I guess, was it isn't that protective. So this chart is a little bit misleading. It only really goes up to 30 here. So these bars look a lot bigger than they are. But kind of the most resistant, if you will, animal by blood was this one great horned owl, right in the range of 25% inhibition. Everybody else had really, really low inhibition, all sub, 15 or 13, somewhere down there. So if we think about those numbers in a different way here, those are the same values, but just shown numerically. All of these have very, very low inhibition of rattlesnake venom. So our chickens are right at 0, which is where we would expect, right? These are a domestic animal and they don't eat rattlesnakes. But then all of our other birds of prey are very, very low on average. So in the neighborhood of 3, maybe to 5, right? So these aren't very inspiring values. And when we think of 3 to 5 and we compare that to rodents, we remember that our wood rat had inhibition values as high as 80%, right? So he was able to neutralize 80% of that venom's activity. So much, much, much greater than any of these birds of prey. Now, what does that mean? So I guess in short, it just means that these raptors, at least the raptors that we worked on, do not appear to have an endogenous protection mechanism against snake venom metalloprotease. So what that means, even more distilled down, is that raptors are really sensitive to rattlesnake venom. So if they get bit, they are going to be in trouble. And we see evidence of this all over in the literature. Myself and a couple others actually published on this in 2021 with some case data. But this hasn't been a surprise, right? In 1994, this group here published on the apparent fatal snake bite in 3 different hawks. So we know that snakes can kill hawks, but we also know that hawks continue to eat snakes, right? So how are they doing this? Well, raptors may have these physical or these behavioral adaptations that guard them from envenomation. And we talked about physical, right? So things like feathers, stiff feathers that can keep fangs off of your body. Their feet are very hard and the blood flow is very interesting down there. So there's some potential for venom localization. So if you're bit in the foot, maybe you can keep that injury to your foot and prevent it from spreading to your body. And then finally, these predatory displays, right? So here's a bird kind of doing a wing display, and it'll presumably be very careful grabbing that snake behind the head. So maybe that's what they're doing. Now, future work, I think there's so much more to be done here. So what safeguards do these specialists have in place, right? Whether those are physical, behavioral, or maybe they are actually inside the raptor. So here's a laughing falcon, and it's eating a highly venomous fertile ants viper. And then here's a secretary bird eating a very venomous puff adder. So these birds are doing it, right? And they're doing it frequently. So how are they able to do it? All right. So I'm going to do one final pivot here on you guys. I'm sorry. I hope we're keeping up. I know there's a lot going on. All right. And this last pivot is kind of where I'm at now in the direction that I'm moving in. And that's snake biodiversity, venom, of course, and then a little bit of human health. So this is kind of what I'm working on now. And I'm for now branding it just this integrative approach to understanding venomous snakes. This is work I'm doing at Oklahoma State University. So a lot of it is in progress. All right, so our lovely quote, right? I come back to this quote a lot. And I think the key thing here is that we're not dealing with the depths to which evolution can sink. Snakes are really interesting. And I hope that I've kind of convinced you of that at this point with all of this banter that we've had. So maybe we've been feuding with snakes for longer than we thought, right? Maybe it's not just AF Scutch. Maybe it's not just your uncle who killed a snake with a shovel one time. Maybe snakes and people have had a longer conflict. And I think there is evidence for this. So if you want to read about this, this book is wonderful. I would recommend checking it out. But there's this idea that snakes and humans have a really, really long, complicated relationship. And some evidence for this comes from our spitting cobras. And when we really dig into understanding the evolution of why spitting cobras spit venom, it seems to line up perfectly with our own evolution. So when primates in Africa began to stand up and they began to become more human-like, that is when cobras in Africa started to spit. And they didn't just spit once, they actually spit twice in evolutionary time. So some really strong support there. And then conversely, when these upright kind of human, early human predecessors moved to Asia, that seems to line up perfectly coincidentally with when cobras in Asia began to spit. So maybe spitting venom is directly something that evolved for human snake interactions, right? Or primate snake interactions at the least. And then additionally, there's this mountain of evidence that babies in particular, so really, really young humans, and then of course, really young other primates, baby monkeys, baby great apes, they're able to recognize snakes preferentially. So if we show them pictures of 100 different things and one of those is a snake, they're always going to be able to pick out that snake. And we call that snake detection theory. So there maybe is some evolutionary wiring on our end and on the snake's end for this little feud that we've been sharing. So keep that in mind. And that feud continues today. And it continues via snake bite, right? So we don't think about this living in North America, but it is a really big problem in the developing world. And snake bite kills 100,000 people yearly, right? So here's somebody, working in their field, probably in India in this particular case, and there's a cobra in their field. They didn't ask to be put in this position, and neither did the cobra. But because of that, they are susceptible to snake bite. Now, of course, 100,000 people dying a year is, a horrible tragedy, but the real burden of snakebite falls on disfigurement. So an additional 400,000 people every year are permanently crippled by snakebite. And, this might be the loss of a finger, as you can see in the image there. It could be the loss of a whole limb. But the problem is that when your livelihood depends on manual labor, right? So you're maybe picking a crop or you're making a product or something like that, this loss of limb could mean permanent unemployment, right? And because of that, snakebite can actually drive the cycle of poverty in the developing world. So not something we think about at all, but a really, really huge problem. And this is turning the snakes that we love, or the snakes that some of us love at least, into a bigger problem than they need to be, right? All right. So because of this, obviously, there's been a lot of global attention on snakebite, and the World Health Organization has really kind of honed in on this as of late, and people are starting to think about it. So that's obviously really good for people, and it's really good for snakes. So why is snakebite such a big problem, right? Why can't we just go and get an antivenom like we do here? Well, there's some barriers in place, particularly in the developing world, and I'll talk a little bit about them here. So one major one is this reliance on traditional healing. So a lot of people in these developing areas aren't really familiar with Western medicine, right? And traditional healing might be as simple as, bandaging a piece of charcoal to the bite, you taking a piece of glass and cutting out the bite site. These aren't actually doing anything, and they often make things even worse. So this is not good. But we also have an issue with antivenom availability and accessibility. So if you're, not making very much money, you live in the middle of nowhere, can you even get antivenom? Can you afford it? And then if you can afford it, does it even work? So this is kind of where I'm interested. Does the antivenom that you're going to get actually work for you? All right, venoms, right? Same slide, we saw this. Venoms are complicated, and they actually vary across the landscape. So here's one type of snake. This is a Mojave rattlesnake. And we see that we have several different venom types across the landscape, right? 4 different venom types, and this is all one snake. And this variation can happen due to a bunch of different things. So genetics, climate, diet, all of that. But that makes it very hard to build any one antivenom that works well. So let's think about 3 steps in snakebite. So snake diversity, right? How are snakes related to each other? We can think about then venom variation. So how does venom vary even within a single species? And all of that can inform us on how to build a good antivenom. All right. So I work on the monocled cobra right now. And this is a cobra that's, quite familiar. And it's distributed across a lot of South and Southeast Asia. So here's some cobras kind of at a snake pit attraction in Thailand. People are very familiar with this species. So let's think a little bit about diversification. What do we know about this one cobra? Well, again, this is the range of the cobra. These red dots are just some of the genetic samples I have right now that I'm working with. And we know that cobras are complicated. That's one thing we know. So here's some work from the 90s that kind of tried to split up all of the different cobras in Asia into species. These were all previously considered one species of cobra, right? But fortunately, this work in the 90s really helped to delimit some of that. And this work used museum specimens, right? It counted scales, it looked at coloration, And museum specimens are great for some things, right? You might be able to count scales on this specimen, but some specimens in museums look like this. And this is a pair of boots. It was confiscated. It's made out of cobra. There's not a lot of data you can get out of this. So while some of these assumptions might be really, really good, some of them are also not great. And that can be due to the data quality. So nowadays, we rely a little bit more heavily on genetics. So if we think about genetics and we think about the previous genetics work that has been done with the monocled cobra, here's an earlier tree from 2016. There's a lot going on here. But again, the take home here is that this monocled cobra in Bangladesh and this monocled cobra in Burma appear to be relatively unrelated to each other. So there's maybe a suspicion that we're dealing with two species of cobra that are actually nested under that one group currently. All right, a little bit more work came out and we find some other weird stuff going on. So this paper actually found the monocled cobra to divide into 4 distinct groupings. So potentially as many as 4 unique species in this monocled cobra. And then again, we see a division here into 2 species. And you can see that these 2 kind of pinkish purplish branches fall out pretty far apart. So these 2 cobra lineages are not very well related. All right, and then bam, 2022, stuff really came to the forefront. And this group out of China described a brand new cobra. So it's 2022 and we are still describing new biodiversity. So this guy is a brown banded cobra. It is not the monocled cobra, which it was previously considered to be. And this group found it to be present in a lot of southwestern China. All right, so do these snakes look different? I think so, right? They do look very different. One has stripes, one doesn't, one's darker, one's lighter, but these are both considered to be monocled cobras. All right, so the thing going on here is that the monocled cobra, air quotes, right? The one monocled cobra might actually be the monocled cobras plural. So we see evidence coming out that there are potentially other species in this group. We obviously have that new description, that new cobra from 2022. So keep posted here. I think there'll be more coming out. But the jury's out. We got to do the work and find out. All right, so let's think about venom variation. So we know a little bit already about monocled cobra venom. So just a quick refresher there on our Mojave rattlesnake. But monocled cobra venoms are highly variable at the geographic level, right? So cobra venoms in Malaysia do not look like cobra venoms in Thailand, and neither of those look like cobra venoms in Vietnam. So we kind of expect that, right? Big spatial scale venoms look different. But it gets even more confusing because monocled cobras are even variable within one population. Now, these are 2 snakes from India. And you can see that just the colors on the pie chart are all over the map, right? So we see strong variation even within a single population. So that gets difficult. So then the question is, how does this variation in venom correspond to the evolution of cobras? So how does relatedness and how do those 2 things work with one another? We don't know. We're working on it. All right, and then finally, that antivenom and clinical management. So antivenom is the only effective treatment against a snake bite, right? And there's just a quick infographic about how it's made. But then the big question is, what if the venom of the donor snake, the snake that we're making that antivenom out of, what if it's mismatched with the biter snake? Now what happens? Well, this is a problem. And spectacled cobras or related cobra, they have this problem. So if you're bitten by a cobra in India versus if you're bitten by a cobra in Sri Lanka, these are the same cobra, but we actually need 2 different antivenoms to treat these bites because these venoms are so different. And we see this same thing happening with our monocled cobra. So the Thai venom and the Thai antivenom correspondingly, that product doesn't really work in India. It's not efficacious in India, even though we have the same cobra. So what other gaps might be present in this antivenom coverage in monocled cobras? Again, we don't know. We should probably try to find those, right? That's pretty important. All right, so kind of wrapping up here, the main take home, I guess, out of all of this is that to really save snakes and protect snakes, we need to fix snake bite, right? We need to save people and protect people. So snakes are important. They're important parts of the ecosystem. They're important for pest control. And of course, there's all kinds of good medical things that we can pull out of their venoms to help us. But to understand snakes from the ground up, right, to really understand what they do and how they do it, this can only help us reduce that global toll of snake bite. So snakes are important. We need to keep them around. And then just the last thing here, not knowing stuff is really, really cool, right? There are so many snakes out there. Here's just 2 quick pictures that we don't really know that much about. Here's a couple more pictures. And we owe it to ourselves, right, to learn all this cool stuff. People like us have to go out there and play with snakes and find out. So the next time you're walking through the woods, I want to encourage you to flip a log, flip a rock, see if you can find a snake. Take a minute to learn about it. If it's safe to pick up and you're comfortable, maybe pick it up, make a snake friend. The key point here is that we just have so much to learn, and it's going to take a lot of people to learn all this stuff, right? So I want to encourage you guys to be those people. Go out there and learn something about a snake. All right. So with that said, that's all I've got. Sorry, I know that was a lot and you guys bared with me, so I really appreciate it. But I would be happy to take any questions at this point.
Okay, so we're gonna start asking questions. So anyone viewing, if you guys want to type a question in the Q&A, feel free to.
Okay, I also did something for you guys. You guys asked me to do this last year, so I brought a snake with me. So this is her name is Puppy. We call her Puppy. Water Puppy is the but she's a green anaconda. So this is a young green anaconda.
Wait, just help this video. Ask him.
All right, so I'm just gonna put her right around me, and I'll take whatever questions you guys have. Go for it.
All right, so everyone here is very interested in the snake.
Yeah, I'm sure. Yeah.
We're sure that's safe.
I'm pretty sure it's safe. Yeah, I know a little bit about snakes, and I'm okay so far.
What's the snake's name?
So her name is Puppy. A friend of mine named her Water Puppy. So she's a green anaconda. This is the world's biggest water snake, right? So these guys get huge, and they're almost fully aquatic. So they live underwater.
Wow.
So yeah.
All right, so with the first question from Channy Mitchell from Elgin High School. Can you explain the difference between toxins, poisons, and venoms?
Absolutely. So that's a great question. So toxins are kind of the broad category, right? So toxin is anything that's toxic. Poisons and venoms are a little bit different. So a poison is going to be a toxin that comes to you via inhalation. So you breathe it in, or maybe you eat it, or maybe you get it in your eyes or something like that. So it's going to go through a mucous membrane, right? Whereas a venom is a toxin that gets injected into you. So a snake bites you, that is a venom being injected. Some fish are venomous, some mammals are venomous, but venom almost always involves an injection into your bloodstream.
Okay, so next question from Maria. How many mice can a snake eat in a day?
Oh my gosh, that's a great question. So it depends on the snake. So I mean, some snakes are very high metabolism. Something like a rat snake or a racer could maybe eat, I don't know, if it was really hungry, maybe 10 or 15 mice in a sitting. But then you have to remember that snakes generally have a low metabolic rate, right? So it'll have a big meal, it's going to sit for a while, and it won't need to eat again for a while. So it could eat a lot in a day, but they don't need to eat very often.
Next question from anonymous attendee. What was your course of study in school?
That's a great question. So I grew up in Canada, as I mentioned. So I went to high school there, and then I did my undergraduate degree also in Canada. So we didn't have any reptile people there. I had to work on birds, which was not, really my plan there. But I worked on birds, and then I ran off to the States. So I did a master's in Colorado, where I did the rattlesnake stuff, and now I'm working on my PhD here in Oklahoma, where I'm doing the cobra stuff. So it's kind of been all over the map.
Okay, Neil, could you stop sharing your screen for a bit?
Yes,
so I
can. Let me see if I can turn that off. Can you share?
Thank you so much.
Of course.
Thank you.
All right, I think we're stopped. Perfect.
Next question from Maria again. Have you ever been bitten by a snake?
Yes. So I get bitten all the time by snakes. It's just part of the job. I've never been bitten by a dangerous or venomous snake, but you have to remember these things are tiny, right? They're usually terrified. They're scared for their life. So the only real defense, they have two, right? So they can either poop on you or they bite you. And usually if they're really scared, they do both. Snake bites don't hurt. I mean, I would get bitten by a snake any day over a hamster. A hamster bite is horrible compared to a snake bite. So if that is any metric for you, take the snake bite over the hamster bite if you can. Yeah.
Laurel Hill, do the mice die quickly or slowly when bitten?
So it's variable, right? And it depends a lot on the venom. So some venoms are made, I'm going to air quotes here, they're made to kill mice very, very quickly. Some venoms are not meant to kill mice quickly, right? So it just depends on the snake.
What type of snakes are like better at killing, have the venom to better kill them mice?
Yeah, so it's all over the map. So in general, venoms that are more paralytic or neurotoxic, so things that work on the nervous system or on locomotion, those are going to kill a mouse or at least stop a mouse a lot quicker, right? So maybe a Mojave rattlesnake, maybe a cobra, whereas some of the vipers have that more degradative venom, which will break down the mouse. It'll cause a lot of bleeding, but it won't die very quickly. So it's probably a painful death. Yeah.
From anonymous attendee, is it true that if you hold a snake by the tail that it cannot turn and bite you?
That is not true. So do not do that. Yeah. So a lot of people think they can, wrangle a rattlesnake by picking it up by the tail. That thing can absolutely flip around and bite you. So if a snake is venomous, the rule is just don't touch it and you're going to be fine. Yeah.
So is the best way to handle a snake by grabbing the head of it?
So I personally would not. So if you're not, a snake person and you're not really trying to do anything with the snake, there's no need to grab it behind the head. The only time we do that is if we're doing, maybe a surgical procedure or we're taking venom. If you find a snake in the wild and it's, a non-venomous, harmless snake, just pick it up. Pretend you're a tree, pick it up kind of like this, and it'll usually just sit in your hands, right? It might freak out for a couple of minutes, but once it calms down, I mean, it's just going to treat you like a tree and move from hand to hand. Yeah.
Okay, and then from another anonymous attendee, how many people are eaten by snakes each year?
How many people are eaten by snakes each year? I would say, so I am making this figure up. I'm going to say 0 to 1. So snakes don't really eat people very often. So they can, right? And I mean, big snakes certainly could. But the amount of actual verified people being eaten by snakes is almost, it's like I could count it on one hand. Yeah, it's very small.
From Laurel Hill again, how do snakes digest their food and how long does it take?
It's a great question. So it depends on the snake. But for big snakes, which are probably the most interesting, so something like an anaconda or a big python, it's going to eat a really big meal. So let's say like a deer or something like that. And then what happens is it goes under this process that we call upregulation of digestion. So its stomach is going to expand, its heart is going to expand, its intestines are going to expand. And for, a couple of weeks, maybe a month, it's just going to go nuts digesting that deer. And then a few months later, there'll be no more bump, the deer will be gone and it'll be ready to eat again.
Jocelyn asked, what's the rarest snake you've ever seen?
Oh man, the rarest snake that I've ever seen, I'm gonna say probably by population size. So it's this really cool little viper. It lives only on a couple of islands in Greece. It's called the Milos viper. And it's super cool. So it's just a viper. It's kind of a short and squat looking viper, but there aren't very many of them left in the wild.
Oh, what makes that viper so special?
So it only occurs on these couple of islands. And it used to only eat birds. So there weren't really a lot of native rodents on the island. So the birds would migrate through, they'd fly to the island, and that's when the viper would eat them. So it's a really cool little viper.
Okay, from sorry, I can't pronounce that name. Mana Khan asked, what subjects do you suggest taking in grade 11 to 12 to go into a field related to herpetology?
That's a great question. So I hate math. I'm really bad at math. I'll just put that out there right now. Be good at math if you can. So statistics are really, really important if you get into science. So I know they're really boring and they're not a lot of fun to learn, but learn them if you can. I would also recommend just whatever biology stuff is available to you guys, take it, right? So whether it's bird biology or fish biology, it all uses the same kind of methods, the same kind of techniques. So do what you can, right? Take what you can.
What's your favorite snake, or what is your most favorite interesting snake?
So I like all snakes, so that's not really a fair question. But right now, I'm at, right? So I'm obsessed with cobras. Cobras are what I spend probably 90% of my snake energy thinking about right now. But that'll change probably. I mean, I'm all over. So.
Question from Laurel Hill again. How wide can a snake open its mouth?
Very wide. So that's a good question. So everybody thinks they unhinge their jaw. They don't unhinge their jaw. That would probably be really, really painful. There's a little connection right here at the base of the mouth, kind of right where we would have this. So this bone on a snake is not one bone, right? It's not fused. There's cartilage in between. So this part right here can stretch out. And because of that, they can actually get really, really wide, and they can eat something, I don't know, maybe 4 times as big around as they are. But again, it depends on the snake.
What is the most biggest snake you've worked with?
The biggest snake that I have worked with, so it'd probably be a reticulated python. So we bump into them every now and then in the wild when we're doing our cobra work in Asia. And then we also see them in captivity, and they can get, I would say, almost approaching 20 feet is relatively common in captivity. So that's a very big snake. It's a lot of snake to handle and work with for sure.
Why do you think they got that big in that area?
Yeah, that's a good question. So, I mean, snakes run the gamut, right? There's some that are really, really tiny and they live in flower pots, little tiny flower pot snakes. And then there's a handful, like this green anaconda that just get huge. And I think, there's a lot of food out there. So I think conversely, snakes have to evolve and just take advantage of those food options. And because of that, we still have giant snakes that eat giant food.
From an anonymous attendee, I've heard that snakes can control how much venom is injected. Is this true?
Yeah, that is true. That's an awesome question. So they can control that. We call that venom metering. So if you want to read into that, just Google venom metering. There's a ton of literature on that. But a lot of people think like a baby snake is more toxic or less toxic. I mean, I hear all kinds of stuff. That's not true. Snakes can control how much venom they're injecting. They know how much to inject. Yep, there's a lot of thinking that goes into that.
They also asked that they've also said that they've heard that younger snakes are more venomous than older snakes. Is this true?
That's a myth that goes around a lot. It is not true. So younger snakes don't have as much venom, right? Because they're smaller. And again, they know how to control what they're injecting into something. So they're not any more dangerous.
What patterns do you notice with snakes and their venom?
What patterns? That's a really good question. So I don't know if there are necessarily any really great patterns that really jump off right off the top of my head. There are, some patterns that come back to food. So things that eat lizards, things that eat rodents, things that eat birds. There are some differences in venom that we might expect. But I think outside of those kind of general relationships, venom can be all over the map. And that's what makes it so much fun to study.
How do you, by Balchan, how do you treat a snake bite? What are some of the things you need to do?
I think that might be my mom. I think she's in here. Anyway, how do you treat a snake bite? Well, mom, the first thing you do is don't get bit. That's step 1, right? Anyway, so if you do get bitten, right, don't do anything at home. There is no home remedy. Just go to the hospital. In the US, we're very, very lucky. There's a lot of really great products to treat snake bite. So if you're bitten by a snake here, go to the hospital. They'll give you antivenom. You'll probably be home in a couple of days at most, and everything should be fine. So don't cut your arm off. Don't put a bandage on. Just go to the hospital. Yeah.
So from Laurel Hill, can snakes eat prey significantly larger than they are?
Yes, that's what they're good at. So that's one of the things that snakes are really, really good at, right? So again, they can't chew, they can't break prey into smaller pieces usually, but they can stretch, right? So an adult green anaconda like this, when this snake is fully grown, I mean, it can eat like a white-tailed deer, like a full-grown white-tailed deer. And that's a meal that will keep it going for months, right? That's a meal that could keep it going for the whole year. So snakes are good at this. That's what they do.
Is it true that pigs eat snakes? Can they eat venomous snakes?
They do. So lots of things eat snakes, which is crazy. Pigs are one of them, right? And venom. So we talked about venom and poison. You can eat venom. I mean, if you wanted to, you could eat venom and it would be okay, right? So venom needs to go into your bloodstream to cause any problems. And 90% of the time, what that means is that if you eat it, you're just going to digest it. It's not going to cause any problems. Don't eat venom. I'm not encouraging that. If you have a cut in your mouth, you will definitely get envenomated. But if you're healthy and your digestive system is normal, venom doesn't hurt you.
Okay, so I think our final question will be again from Laurel Hill. How much venom does a snake have, and how much does it take to kill a human?
Yes. So again, great question. Very, very variable. So a big snake, like a big rattlesnake, might actually have a couple milliliters of venom. So maybe 3, maybe 4 milliliters of venom in a really big rattlesnake. And again, how much venom does it take? Totally depends on the snake, right? For some, it could be like a drop, like a tiny micro amount, and then for some, it might be a lot. So all over the map. No good answer there.
What is the best smaller size snake to have as a pet?
Pet, okay, I get this question a lot. So there's a lot of really good pet snakes out there. I think the common ones are probably things like ball pythons or corn snakes. They make great pets, you know, king snakes. Yeah, really any smaller snake that you could find would be good. Yeah.
All right, so I think that's all the time we have. Just want to make one quick comment. And Balchan did confirm that it is mom, so.
Knew it. I knew it.
That
Lurking in here.
was
Yeah.
a comment we got. So, Walker, thank you for your time and being able to be here and give us that great presentation. And thank you for answering our questions.
Thank you.
Perfect. Thank you so much, guys. Thanks for coming and hanging out.
Goodbye, puppy.
Right. Okay. See you guys later. Thank you.
Oh, it was great.
Bro, stop it.